Welcome to the presentation of the 24th Annual Gruber Genetics Prize. This prize honors a leading scientist or scientists for groundbreaking contributions to the realm of genetics. We're very pleased to host the Gruber Foundation, and I invite their executive director, Sarah Rea, to come and tell us a few words about the foundation and the prize.
Thank you, Professor Wolfner. We are delighted to be here in National Harbor to present the 2024 Gruber Genetics Prize at the Allied Genetics Conference. And I know they're trying to change the PowerPoint behind me, so I'm stalling a little. While we are here to honor Hugo Bellen, let me mention that he will join still unselected cosmology and neuroscience recipients on our 2024 roster. If it comes up, you'll note our 2023 laureates are still on. Please note that nominations to the 2025 Gruber Prizes are open until December 15th, 2024, and that we encourage nominations that reflect the diversity, the breadth of the field and the diversity of those working within them. Before we return to genetics, I simply must acknowledge our co-founders, Peter and Patricia Gruber, whose combined vision and leadership established a national prize program. Care doing so gave it the legs to stand on its own. I invite back Mariana Wolfner to say just a few words about our collaboration with the Genetic Society of America.
Thanks, Sarah. GS is foundation to nominate the members of the Election Advisory Board. Honor the groundbreaking contribution of Dr. Franklin and to inspire and support new generations of women in the field of genetics. GSA administers this exciting fellowship, which the Gruber Foundation generously funds. Thank you.
Thank you again, Professor Wolfman. Our relationship with GSA is invaluable, and we appreciate being hosted here today. Hugo Bellen joins an illustrious laureate list, and he was chosen by a distinguished selection advisory board. We sincerely appreciate the knowledge, commitment, and enthusiasm that these advisors bring to the judging process. Several of them are here with us today, including our chair, Alan Spradling, who I now invite to present the official citation to the prize and introduce the scientific of our recipient. Professor Spradling.
Thank you. The Gruber Prize Selection Committee, I'm tremendously pleased to introduce the winner of the 2024 Gruber Prize in Genetics, Hugo Bellen, of the Nathan Neurological Research Institute at Baylor College of Medicine. Hugo Bellen has been a ubiquitous figure in the field of Drosophila developmental genetics and neuroscience for more than 30 years. Today, if you work with Drosophila for even a short time, you will find yourself using strains that were produced in Bellen's laboratory. That's because Hugo long ago appreciated that progress in biology and medicine is largely limited by the sophistication and power of its genetic and genomic tools. He consequently devoted a great deal of his time and thought to creating or upgrading many of the genetic resources widely used by our community. For example, Hugo played a major role as a postdoc in developing enhancer trapping technology that makes Gal4 lines so powerful. His lab developed the Pac-Man system of large genomic clones that can be manipulated by recombinering and transferred into flies for diverse purposes. He generated systematic collections of duplication strains and new methods for rapidly mapping EMS-generated point mutations. As a leader of the Drosophila Genome Project, he helped conceive and generate, largely in his own laboratory, the widely used system of mimic insertion lines located throughout the genome, including in repetitive and heterochromatic regions. Mimic lines allow the precise swapping of arbitrary DNA cassettes, allowing complex, precisely predictable genomic structures to be routinely generated virtually anywhere in the genome. Working with Norbert Perry-Mohn's lab, Bellen has recently extended the collection of targeting sites analogous to using analogous constructs based on CRISPR. He raised the funds to carry out many of these projects himself, and he has served on countless review panels and committees dealing with genetics and genomics for science agencies, private philanthropies, and scientific societies, including serving as GSA president. He provides the Bloomington Stock Center and many other worthwhile resources with invaluable advice through his long service on their oversight committees. Perhaps the fullest measure of why Hugo Bellen so richly deserves this award, however, is his efforts to expand the role that model organisms play in medicine. He has brought to fruition, step by step over many years, the technical capability to use engineered Drosophila as precise models for specific patient disease mutations. As a young scientist, Bellen studied neurodegeneration mutants in Drosophila to decipher the biological principles of how neurons and glia function and interact. Few in the field felt that such basic science could be directly applied to the much more complex human nervous system or to the many patients burdened with neurodegeneration. However, as advances in genomics revealed the tremendous conservation of genes and pathways among all animals, Bellen became convinced that many human genetic conditions are caused not by disruption of genes whose function has changed during evolution, but by genes whose function have been conserved widely in animals. He became an early participant and strong advocate of the Undiagnosed Disease Network and its Model Organism Center, which helps families around the world whose child have been born with an unknown and rare genetic condition. Essential to his success in advocating for this approach has been his ability to rapidly identify and validate likely pathogenic variants. Equally important has been Hugo's ability to communicate clearly, rapidly, and transparently with physicians whose responsibility is to care for patients with problems of unknown etiology. While many fine scientists and physicians are helping to advance the UDN approach using multiple model organisms, Hugo's contribution to the astonishing growth and progress of efforts in this area is second to none, and he is widely recognized as a preeminent leader. The award citation states, the Gruber Foundation proudly presents the 2024 Genetics Prize to Hugo J. Bellen for ushering in a new era of model system genetics with direct impact on human disease. Bellen developed novel techniques that allow human proteins bearing patient mutations to be expressed under the normal regulation of their Drosophila orthologs. Working in close collaboration with clinicians and basic scientists, Bellen and his partners have elucidated dozens of rare undiagnosed neuronal development and neurodegeneration syndromes, providing new examples of how rare diseases can illuminate underlying mechanisms of more widespread conditions. Bellen exemplifies the highest standards of quality, collegiality, and generosity in producing and widely sharing innovative genetic reagents and genetic tools that have transformed Drosophila genetics. I now invite Hugo Bellen to the podium to receive the Gruber Prize.
Before I start this talk, I would like to kind of acknowledge the importance of people that have helped me to get here. And first, I would like to thank Peter, Patricia Gruber, and their foundation for obviously supporting genetic prizes and promoting genetics. I think that's really been important and providing such a generous award. Their visionary approach is greatly appreciated, and their staff, Sarah, Rhea, and Stephanie Ratton, have really made this a pleasure. Second, the Gruber Foundation, together with GSA, have assembled a panel of very good and prominent scientists that, if you look at the people that they've selected in the past, have really done a great job at selecting people that have had a major impact in the field, and I feel very honored by being selected. Third, you don't get an award if there's not people that are nominating you. And so I have to really thank people that have nominated me for this award, and the colleagues that have written the letters to support these nominations, because they're really important, and some of them are here in the audience. Fourth, an enormous amount of depth and gratitude to my spouse, Catherine, who for 40 years has really supported me in doing this work and standing behind me. Without her, this would not have happened. Fifth, the members of the lab have obviously played a determinative role in the success and accomplishments of our lab. And I'm glad to see that today there's quite a few people that worked with me 30 years ago, including Troy Littleton from MIT, Mansour Bhatt from UT San Antonio, and Bassem Hassan from Paris, who specifically flew over for being here. So the support of my former trainees is really valued. And obviously, the work that we've accomplished in the lab would not have been possible if there weren't 100 or close to 100 trainees, postdocs, graduate students, and many technicians that have helped us achieve what we've done so far. Finally, I obviously have to thank the funding organizations, and the Howard Hughes Medical Institute has supported my lab for many years and has allowed me a lot of freedom to do what we've been able to do. And in the last 7-8 years, the NIH has stepped up tremendously and provided a lot of support for the work that I'm going to be covering today. Finally, I want to thank the Bloomington Drosophila Stock Center Flybase and the Drosophila Genome Research Center, because without them, much of the work that we would have done would not have led to almost anything. So thank you. Today, I'm going to be talking about, I need to find, oh, that's not the right presentation. It's on its way. All right. Great. So today I'm going to be talking about the study of rare pediatric neurological disorders in flies and how this has driven discoveries in common disease. And so the goal of the work in the lab is really to advance basic science as well as medicine by studying human genes associated with diseases in flies. And this is a collaborative project where we try to promote efforts among geneticists, human geneticists, and other scientists, first to help in the diagnosis of rare diseases. There are millions of people with rare diseases, and many of these rare diseases are caused by mutations in genes, and identifying these genes is obviously the first step to thinking about any therapy. We also want to help elucidate the pathogenic mechanisms of these diseases using fruit flies, and have been quite successful at doing this. And this then allows us to select targets to help us in the discovery of targets for drugs. And given the speed at which we can work with fruit flies, that has also been effective. And in essence, we like to promote the discovery of functions of genes that have been poorly characterized, because quite a few of these genes that cause human disease, when you start looking at the fruit fly homologs, you notice that they've not been very well characterized. And so to participate in this endeavor, we decided to apply for funding from the Undiagnosed Disease, and happy to be selected to participate in this endeavor. In essence, the coordination center of the Undiagnosed Diseases Network in Harvard gets applicants, that is, people that have problems. And many of these people have been through a diagnostic odyssey. They've gone from one doctor to another. Most of the time when they apply, they've been 5 to 70 years already undiagnosed. And it typically takes about 10 years for these patients to become diagnosed. Once they apply to the coordination center, they are assigned a clinical facility, preferably in their vicinity, where they can then go for a couple of days and be kind of phenotyped by the physicians to try to see if they can identify the cause of the disease. Very often they end up sequencing the parents and the kids, or the probands and the sisters and brothers, and try to identify 3 or 4 individuals from the same family, so they can sequence those and see if they can find a possible lesion, genetic lesion, that may be the cause of this disease. And when they do so, sometimes they find 1, 2, or 3 genes that carry variants that they then submit to the Model Organism Screening Center. And in the Model Organism Screening Center, what we've done is we bring physicians together with model organism researchers, Drosophila, C. elegans, and zebrafish, and we brainstorm the variants that are submitted. We developed this website, Marvel, where people can query all this information from all these databases, from all these model organisms, as well as all the human information, and try to select variants in genes that may indeed be the cause of the disease. And we're not always successful. We probably fail about 40 to 50% of the time, but we're quite successful for the other 50% of the time. And we assign then the genes depending on the knowledge and the variants and the interest of the groups to any of these 3 model organisms: worms, flies, or zebrafish. In the fly core, we do about 50% of all the genes that are submitted. And the reason why we can do so many genes is because we developed technologies that have really allowed us quickly assess a lot of information related to the genes and the variants in the human. And I just, with this slide, want to kind of introduce you to what we do. So through the website developed by Norbert Perrimon in Harvard and the diet score algorithm, we can identify orthologs of the human genes in flies. And we can then insert this cassette that is shown here, the splice acceptor T2A GAL4 cassette, in the gene. And the purpose of these insertions of this cassette is to disrupt the gene and create a mutant. And 90% of the time, we create a very severe loss of function or mutation or null allele. But by inserting a T2A GAL4, what we do is we produce the GAL4 protein in the proper spatial and temporal expression pattern of the gene. And by doing so, we now have a tool to do an integrated study of the function of the gene. The first is we can use the GAL4 to drive a US nuclear M-cherry and find out where the gene is expressed. This has been very variable. For only a few % of the genes, maybe 10% of the genes in Drosophila, if that much, we have antibodies. And so being able to determine at all stages where the gene is expressed has been extremely valuable. The second thing that we can do is obviously determine the loss of function phenotypes and find out what are all the loss of function phenotypes that are associated with this gene. And the third one, which is also very important, is we can drive a US human cDNA to rescue the fly gene. So we've collected large libraries of full-length cDNAs, human cDNAs, and as I'll show you in the next slide, insert that into fruit flies, and that allows us then to systematically see if we can rescue the mutations in the fly with the human reference cDNAs. And if that's the case, we can test the variants, and we have an in vivo test tube, if you want, to test human variants in a living organism. As shown on the right here, or the GFP allele, we can also generate a insertion where we bring in a GFP, and unlike when we do the T2A Gal4, we do not want to create a mutation. What we want to do is create a tag protein with GFP, and we do this typically by inserting an exon in a coding intron of the gene, as is shown here. And this then leads to a GFP fusion and is actually a better strategy than tagging proteins amino or carboxy terminally, as many of you do. We've shown that this is often less detrimental to the function of the gene. And this then allows us to do protein localization studies and find out if it's a mitochondrial protein, ER protein, whatever. But also do loss of function studies, because we can now use the degradFP system to selectively remove the protein in any tissue and do this in a reversible manner. So we can remove a protein and bring it back and assess after a week when we've shown that the protein that's GFP tagged is lost. And then we have a phenotype. Can we suppress the phenotype by returning the protein function? Using the system with the degradFP. And so that's been a very effective system. The combined system has been very effective to relatively quickly study a lot of information about these genes. I want to expand a little bit on the work and the Kanja, a postdoc and currently a junior faculty in the lab, has played really a major role in developing these technologies. And what is shown here on the slide is a gene in which, through CRISPR, we insert this cassette. And what's nice about this cassette is that the homology arms can be very short. And so we have these 100 nucleotides homology arms. And the reason why they are so short is because the cassette is put into a vector, and then we cut out with CRISPR technology the donor construct to make a double-stranded donor construct that very efficiently can integrate into the gene. And at this point, we've reached about 85 to 90% efficiency from design to creating a fly. And so that's really, really important, because that allows us to really tag thousands of genes, and we've currently tagged already 3,500 using this strategy. And the hope is to do this for the rest of the genome. The strategy, as I mentioned, has many applications, but I want to go back through them. The first one is it's a mutagenic cassette, because there is only one splice acceptor site and there is no splice donor. So you truncate the protein, and 90% of these, as I already mentioned, are strong mutagenic insertions. A nice thing is, because the cassette is flanked with FRT site, is that you can express the flipase and remove with the flipase the FRT cassette. And people always say, well, Hugo, you got to be careful. You're inducing with CRISPR these mutations. How do you know that the phenotype that you're characterizing is not due to a second site mutation? The answer is simple. We cross it to US flipase. The cassette is excised wherever the Gal4 is expressed, and the phenotype is reverted. At that point, you really know that your genetics is clean. In addition, this can also be used to excise the cassette in a targeted fashion. So I'll show you an example later where a gene is expressed in neurons in glia. But by using a riboflippase, you can selectively remove the cassette in the animal, just in the glia, and assess what the phenotype is when the glia are wild type and the neurons are mutant, or do the reverse, obviously. And so the other advantage of this cassette that we insert with T2GAGAL4 is that we can replace it with a splice acceptor GFP splice donor, as I already showed in the previous slide, and revert the phenotype that was induced by the T2AGAL4, because this cassette is not supposed to be mutagenic. The GFP is functioning as an exon that is used during the splicing event. And finally, we can use the method to express the GAL4 in a proper spatial and temporal expression pattern and rescue with the fly cDNA, the human cDNA, a human variant, or determine the expression pattern with a fluorescent marker. So by integrating all these properties, we have a powerful tool to study these homologs of these human genes. Key, obviously, is that we need a library of human cDNAs to do this systematically for all these genes. Yamamoto and Susan and Toshi Takano Shimizu have helped us generate this library. So we've collected a library of full-length human cDNAs and make vectors, UAS vectors, in which we integrated these full-length human cDNAs, and then we're able to make transgenes with those, and we can now cross these to the T2AGAL4 lines to systematically see if we can rescue the phenotype. So we've generated with Suselniker 9,000 US cDNAs, human cDNAs, for most of the important genes that are evolutionarily conserved. Made quite a few transgenes, and these are distributed by the Kyoto Stock Center and by the Bloomington Stock Center. And they also allow us, obviously, to overexpress human genes in flies. And combining this with the previous strategy, we've been quite successful at identifying that cause human disease. In fact, this slide summarizes the work from the last 7-8 years, where in collaboration with the labs of Shinya Yamamoto and Michael Wangler, we've been able to identify 50 novel human disease, rare human disease genes. And we're currently working on 40, so that gives you an idea of the scale at which we can do these experiments. What's also important is that many of the postdocs that are doing this work realize that many of these genes are really poorly characterized. And they start digging into the mechanism and trying to figure out really what's going on. And by doing so, they find targets, and some of these targets are substrates for FDA-approved drugs. And that allows them then to relatively quickly use these FDA-approved drugs and test them in Drosophila and see if they're effective. And the ones that are listed in blue here are drugs that they've shown to be effective for the loss of function of these genes. And this list is growing actually quite rapidly. It's a targeted approach, but it's been shown to be efficient. And some of these drugs are used in patients already because they are FDA approved. And some of them have been shown to be very effective. And so I think if you think about the time scale, this has been a productive strategy. Another feature is that these genes that are listed here, some of these genes, have led us to an understanding of much more common disease. By studying these diseases like the gene here that is shown as phospholipase A2G6 or ACOX1, are genes that have led us to an better understanding of much more common disease in the case of ACOX1 for multiple sclerosis, and in the case of phospholipase A2G6 for Parkinson's disease. And I'm not going to elaborate on these two genes and why they have led us to a better understanding, but I'm going to pick a third disease, and that is Alzheimer's disease. What I'll talk about in the rest of the talk is the role of lipid droplets in Alzheimer's disease and how we think this is really important in the pathogenesis of this disease. And it all started again with the study of rare disease genes. I'll explain how we got to those rare disease genes. But the work was done by Lucy Liu in the lab, Matt Moulton and Lindsay Goodman. And what I'm showing here is not a fly brain, but a model of a mouse brain where there is lipid droplet accumulation in the microglia and in the astrocytes. The astrocytes are red and the microglia are blue. And in these mouse, a few weeks after the lipid droplets have accumulated, all the neurons will die. And in fact, these mice are only supposed to live about 70 days. This is a phenotype that occurs after about 3 weeks. So the story started when Xing decided to perform a forward genetic screen where they created 36,000 stocks and screened for 6,000 lethal stocks. So we used very low concentrations of EMS. And they established 6,000 stocks for a screen on the X chromosome using a flipase. So they first screened for lethals, and then they took the heterozygous flies and induced clones in the heterozygous flies. And they screened for two phenotypes. The first one was for the loss of bristles in the thorax. The second was for creating photoreceptors that are mutant and that are normal when the flies eclose, but progressively lose function. There was a screen essentially for a bristle screen in the thorax and in the eye for a progressive loss of function of the photoreceptors, implying that they would be genes that are required for photoreceptor maintenance. In other words, neurodegeneration. And those that are required in photoreceptors for their normal maintenance were then subsequently screened using transmission electron microscopy. And that screen was really productive and very important for the lab, and identified 165 genes on the X chromosome. And of those 165 genes identified on the X chromosome, the list of fly genes is always on one side, and then behind it are the human genes, and 93% of the genes are conserved in human that we identified. And currently, almost 70% of those human homologs have been associated with human disease and are listed in the Mendelian online database, the OMIM database. So the screen was productive and allowed us to identify a number of new human diseases, but by doing it systematically and going to physicians and depositing all these variants and all the genes that we had identified, they identified, Jim Lupsky and Richard Gibson-Bader identified patients and families that allowed us to systematically start studying this. Now you may wonder, you're focusing on a screen here in the thorax and the eye, and half of the genes were genes that affected bristle morphogenesis, half of the genes were required for neuronal maintenance. And so this screen was done, as I said, in the eye, and I briefly want to end Drosophila has been so useful. We're probably all familiar with the work from Jerry Rubin and many other people in the fly community. They've worked on the eye, but from a perspective of looking at neurodegenerative phenotype, the eye offers really a lot of advantages. It's a non-essential tissue. The phenotypes are really easily scorable. They correlate with brain phenotypes, and I'll show you that repeatedly. The Almatidia have a stereotypic structure, and the photoreceptors are surrounded by glia, so we can look at this communication between photoreceptors and glia. It allows us to probe neuronal glia interactions, and this will become very important in the remainder of the talk. They allow us to assess the neurodegenerative phenotype with a functional assay, the electroretinogram, and the transmission electron microscopy. And some feature that is not available for almost any neuron is that we can control the activity of the neurons by raising the flies in darkness or light and modulate the amount of light and therefore of activity that is present. And so this really gives us a number of approaches that are really important and very elegant. And as you can see on the right here, we stain eyes with Nile red, and the Nile red will label these dots, which are the rhabdomeres of the photoreceptors. And on the top panel, you see that when there is lipid droplet accumulation in glia, you get these glia that surround these photoreceptors that are now loaded with red and lipid droplets. And this is the key assay that we use to look at the lipids and the lipid droplets in the brain. So the screen identified mutation of, when we did electron microscopy, as is shown here at the bottom, showed that the glia were expanded. So the glia normally are this blue sliver that surround these photoreceptors, and as you can see, the glia are expanded at the bottom when we do transmission electron microscopy, and they're loaded with lipid droplets. And it's very easy to immediately turn to the Nile red assay and see what would happen in these mutants, as is shown here. And so here is a gene that we named Sicily at the time. You can see the rhabdomeres that stain, because the rhabdomere is a stack of 5,000 membranes, and so it contains a lot of lipid, and Nile red will stain these lipids. But then you can see the glial cells that are loaded with these lipid droplets. Here's another mutation, and here is another mutation, MARV. And the human homologs of these genes are NDUFAF6, MARS2, and mitofusin-2. And in all cases, these genes, these human genes, are associated with homolog is NDUFAF6 and causes Lee syndrome. And when we discovered this Sicily gene, we noted that there were very few, but a few patients in Lebanon that had Lee syndrome. And Lee syndrome is a very severe disease. These kids only survive for 2 or 3 years before they pass away, and they become blind and deaf, and it's just a dismal existence. The other gene that we show here, ATSMET, it causes a disease called a rare disease called ARSAL, autosomal recessive spastic ataxia with leukoencephaly. And the third gene, MARV, is a gene that causes also a rare disease when homozygous, called Dejerin-Sota syndrome. The heterozygous MARV mutations cause a disease called Charcot-Marie-Tooth disease. So what is shown here is that these 3 genes cause these lipid droplet accumulations, and they're all encode mitochondrial proteins. And these mitochondrial proteins play a critical role in the function of the mitochondria and the production of ATP. And when they're affected, they create high levels of reactive oxygen species. And so we have these high levels of reactive oxygen species that we can easily monitor when we stain with BODP, C11 BODP. And you can see that in the control at the bottom in green, there is barely any signal, but in the Sicily mutants, the photoreceptors are super efficient at getting rid of these peroxidated lipids and dumping them in the glia. And so they get rid very efficiently of these peroxidated lipids because there is high ROS. There is, I'll show you in a minute, lipid production. The lipids get peroxidated, get transferred to glia, and they pile up in glia. If ROS is really at the root, then we should be able to suppress the production of these lipid droplets, and that happens is a powerful antioxidant, N-acetylcysteine amide, also called AD4. Also called AD4, and this antioxidant dramatically suppresses the presence of the lipid droplets. You can see here, these are the rhabdomeres of the photoreceptors. These are all lipid droplets in glia. At 40 microgram per mil of N-acetylcysteine amide, you suppress this about 10-fold. That's the production of these peroxidated. You also, at the same time, suppress the neurodegenerative phenotypes that are associated with the accumulation. I'll show you in a minute that these lipid droplets that are loaded with peroxidated lipids that they're neuroprotective, but they're only neuroprotective as long as the glial cells do not get overwhelmed. If the glial cells get overwhelmed with these peroxidated lipid droplets, the glial cells will start being dysfunctional, die, and then the photoreceptors will ensue. So the next question is, this is all in fruit flies, this is in fly photoreceptors, what about mouse? Reiter at the University of Washington and a postdoc in his lab, Alberto Quintana, who had gotten support from a organization that supported research on Lee syndrome and had generated mice for a NDUFS4, which is a gene, again, that's required in the mitochondria for a complex one. And as you can see, when Lucy started staining these brains of these mice, we noticed that there were truckloads of accumulation of these yellow lipid droplets in microglia and astrocytes. And these mice live about 70 days, and this phenotype occurs rather early, around 20, 25, 30 days. And again, in the beginning is neuroprotective. But if you look a few weeks later, all the neurons are gone. So when the system gets overwhelmed, the cells die. Now, is ROS important in inject intraperitoneally N-acetylcysteine amide? And we could only do this for a couple of days because the drug was too expensive at the time. It was about $14,000 per week for each experiment. So we only did it for 1 week. But what you can see here is if you treat the mice with 84 by intraperitoneal injection, and you put them on the rotarod assay, the wild type mice and the mutant mice that are injected do well at day 30, but the mutant mice that are not injected do not as well. And very quickly, you see that there is a major problem. In fact, by day 50, the mice can barely move. If you then look at the experiment that was done by another team, and his PI was Vamsi Muta at the Broad, he took the same mice and he did a very simple experiment that I thought was great. The red line shows the mice, the normal lifespan of the mice under normoxia. And so they live about 70 days. If he puts the mice in cages in hyperoxic conditions, they're almost all dead by day 30. And if he puts the mice in hypoxic conditions at 11% oxygen, they never die. So I think this is as compelling evidence as you can come up with to show that ROS is really a critical player in this disease, and that is an important aspect. So then Lucy started being more interested in figuring out what was going on. And in essence, what she found was that the mitochondrial defects lead to elevated ROS production, which activates JNK, which is a known pathway. But what was not known is that JNK phosphorylates SRABP, and SRABP is a key transcription factor that controls in vertebrates the levels of 24 enzymes that are involved in lipid synthesis. And so what you have then is the ROS induces lipid synthesis, and the lipids function as a single reactive oxygen species and transfers those to glia where they're sequestered and they're neuroprotective. When I presented this data before they were published, a physician approached me and said, you should read the chapter from the paper from Alzheimer's in 1907. He says, he documented that there's lots of lipid droplets in glia. So you may want to look at that paper. And I was struck by that because I didn't read the paper, and he mentioned it 3 or 4 times. He called them lipid sacules. And he showed that, yeah, these glial cells in these patients with Alzheimer's disease have truckloads of these lipid sacules. And that was not described at all till we published our paper in 2015 by any other paper. And I was struck by that and started inquiring why that was. And it was because he used a poor fixation protocol and Nomarsky optics. And no other pathologists use these poor fixation protocols. And if you use strong fixation protocols, the lipids disappear and you don't see these. And so you need to do special stainings. Obviously, peroxidated lipids are damaging, and the neurons have found this efficient way of getting rid of these peroxidated lipids. So Lucy started digging into this pathway and figured out very elegantly that lipid droplet formation in glia is dependent on a whole series of players. The first is lactate. Lactate is produced in glia, and there is a lactate shuttle hypothesis by which the lactate to monocarboxylate transporters is transported to neurons. The neurons then have an enzyme lactate dehydrogenase to form pyruvate, and pyruvate dehydrogenase forms acetyl-CoA. Acetyl-CoA, as you know, is the source, the energy source for the TCA cycle. But the TCA cycle is not working very well, and the mitochondria are not doing a good job. So they produce high levels of ROS, junk, and SRABP. And suddenly you have the perfect conditions. You have all the enzymes that are upregulated that make lipids, and you have the substrate for lipids, acetyl-CoA. And so now you form these lipids, you peroxidate them, and you have to transfer them, obviously, to glia, where they can be sequestered to protect the neurons. And it turns out that that's dependent on apolipoprotein. And in flies, there is an apolipoprotein D, and there's two of them, neural and glial lazarillo, and that is these proteins that are shown here. And so what's happening is that these proteins, if you abolish this protein, glial lazarillo, or ApoD, you have no lipid droplet formation. So the transfer of the lipids to the glia is really dependent on these apolipoproteins. And obviously, apolipoprotein E in human is the most important protein in Alzheimer's disease. The ApoE4 variant accounts for more than 80 or 90% of the genes that are associated with disease. ApoE4 is really found in 40 to 70% of individuals that develop Alzheimer's disease. But there is no ApoE in fruit flies, but there is this glial lazarillo. So using our technology with the T2A Gal4, we killed glial lazarillo and replaced it with ApoE. And ApoE, the reference, fully functional, is fully functional in Drosophila and works beautifully. And I'll show you in the next slide that ApoE4 doesn't work at all in this assay. So what you see here on the left is the photoreceptors and the lipid droplets, where the arrows point to the lipid droplets. We remove one copy of glial lazarillo, and this whole pathway is extremely dose sensitive, 50 to 60%. Removal of almost any protein in the pathway affects the accumulation of these lipid droplets. And so loss of one copy of glial lazarillo reduces the presence of these lipid droplets. Now we use the lazarillo Gal4 to express ApoE2, 3, and 4. ApoE2 is neuroprotective. Alzheimer's disease, and you can see there's lots of lipid droplets in ApoE2, moderate number of lipid droplets in ApoE3, and no lipid droplets in ApoE4. And this correlates directly with the neurodegenerative phenotype that we observe in the photoreceptors. When you express ApoE4, after 10 days, you lose 20 to 30% of the photoreceptors, and there's progressive further loss. So ApoE4 is not at all good in this context with respect to the formation of lipid droplets. So given that ApoE4 is a locus that is very prominent in genome-wide association studies for Alzheimer's disease, we turn that have been implicated based on genome-wide association studies for Alzheimer's disease. And Matt Melton is here, decided to start looking at some of these factors that, are prominent factors and that are shown here. ApoE obviously is very high, but then you see this ABCA7 and PICOM and BIN1 and CD2AP. And so you have all these genes. Can we assess if these genes that are associated in genome-wide association studies, are they playing a role in this pathway? And so the idea was then start systematically knocking out with RNAi these genes either in photoreceptors or in glia and trying to assess how lipid droplet formation would be impacted. And so that's what Matt did, and he went to all these genes that are shown here, BIN1, PCOM, CD2AP, ERPA2, LRP, ABC, A1, A7, etc. And here I show you just the example of the knockout of ABCA1 and ABCA7, genes that have been implicated in Alzheimer's disease, in glia and in neurons. And so if you knock out these genes, these ABCA1 and ABCA7 transporters, in neurons, you see that the glia do not form lipid droplets anymore. That's what's shown here, right? That's the control. You make lipid droplets, no lipid droplets when you knock out ABCA1 or 7 in neurons. But if you knock them out in glia, that is not a problem. So there is a dramatic reduction of this. Now, what happens with the photoreceptors over time? After 5 days of knockout in the photoreceptors, you see a subtle reduction, which is much more significant at day 20. And that's true for both ABCA1 and ABCA7. And so by measuring the photoreceptors and the ERGs, you can see that there is a progressive loss of vision in these mutants when knocked out in neurons and not in glia. And another feature I want to bring up, I mentioned NACA here, NACA, and you can see that after 20 days present on NACA for both of these, there is no phenotype with respect to the ERG. So NACA is neuroprotective. So the next thing is, let's look at these other genes. I'm not going to cover them all because it's too long. Here, other genes, and here the knockouts do not show a phenotype when the genes are knocked out in neurons. The lipid droplets are always there. But when you knock them out in glia, you can clearly see that these genes, APA2A, PICOM, and CD2AP are required. And that is quantified here. And again, when you look at the ERGs and the progressive degeneration, we see that these genes, all three, lead to a progressive loss of ERGs over time. They're all required, they're all genes that are required for endocytosis, and I'll come back to that because obviously you need to secrete the lipids, load them on the apolipoproteins, and then the apolipoprotein need to bind to a receptor, and that complex needs to be endocytosed in glia. All these proteins play a role in endocytosis in glia. The next question was, again, is this conserved in vertebrates? Is this process that we see that is dependent on these endocytic genes in glia required in vertebrates? And we collaborate with Maria Ioanou's lab, who actually started this work at Genelia Farm with Jennifer Lippincott-Schwartz. And what they showed is they developed an assay where they have the neurons that they can load with red C12, as is shown here. And then they stress the neurons and they put them on a layer of glial cells. They don't touch the glial cells, but they put them on a layer of glial cells. They stress the neurons with a medium and they follow over a few days what's happening. And as is shown here, you see that there's lipid droplet accumulations in these astrocytes that are below these neurons. So the lipids are taken up by neurons. When the neurons are stressed, they secrete them and they end up in the glia. And so here, if she knocks out PCOM in these astrocytes, you see that there is a very significant reduction of the amount of these peroxidated lipids. And she's shown that this correlates with a higher an elevation of peroxidated lipids as well. The genes that are associated with GWAS, what about studying phenomena that relate to Alzheimer's disease? And now I'm going to turn to two other players, A-beta 42 and tau. Both players are known to play an important role in Alzheimer's disease. And the goal is to determine how A-beta 42 and tau, both important players, affect the function or interfere with lipid droplet formation in this pathway. So here what we show is that A beta 42 is produced, is secreted in the extracellular space, and it turns out that when the apolipoproteins get loaded with lipids, they start binding A beta 42. A beta 42 has 17 amino acids that are hydrophobic, and so when there is enough lipids in the apolipoprotein, the A beta 42 gets taken up in that complex, and that complex to the apolipoprotein receptor and is endocytosed, and then that complex can go to the lysosome where A beta 42 is degraded, the lipids are extracted from the lysosomes, go to the ER, and then form these lipid droplets. So given that A beta 42 is an important player, we were interested to see where A beta 42 would play a role. And the model would be that based on the biochemical experiments, that A beta 42 should use the same pathway, bind to the same players, and therefore should be toxic and interfere with this pathway when A beta 42 is elevated. And Matt did this experiment, and here, this is one of the experiments. I'm not going to show you the other experiments, but here he looks at the photoreceptors, treats the flies with rotenone at day 1 and day 10, barely a lipid droplet that forms here at day 10. This is a very mild condition. He then turns to rhodopsin gal-4 and expresses A beta 42. Again, at day 1 and day 10, it typically takes about 30 days before there is a strong phenotype. You put the two together, it takes 1 day to see strong phenotypes, and by day 10, half the photoreceptors are gone. So A beta 42 hijacks this pathway, and he's shown that if you overexpress SRABP in the photoreceptors, you can suppress this phenotype because you drive the production of transport into the glia, and you can suppress these phenotypes. So I think this is another key point here that argues that A beta 42 uses the same players to be degraded in the lysosomes. So before I go on and talk about tau, I want to come back and say that we started with a rare disease that was associated with Lee syndrome. In fact, Lee syndrome genes cause these high levels of reactive oxygen species in the brains of these kids. And the gene with Sicily, NDUFAF7, became a GWAS locus for Alzheimer's disease. And there is now 4 of these Lee syndrome genes that have become GWAS loci for Alzheimer's disease. And so I think this makes for a strong point that what we do at the biologic level is directly relevant, and clearly now quite a few of these genes have been associated with AD through GWAS studies. Next, I want to turn to tau, which is the last player in the last few slides. We made again a T2A gal4 crimic insertion in tau, and this created, as you can see, a very severe loss of function, about 90% reduction of tau. These flies have a short lifespan. And Lindsay, who's been doing the work, then showed that this gene is expressed in neurons and in glia. And in fact, tau is expressed in mammalian glia. By the way, tau binds to microtubules and controls the function of microtubules. And when Lindsay kind of looked at this experiment, I thought that was really interesting. What she did is she used a repo flip to remove the tau insertion only in the glia, and it nearly suppressed all the phenotypes. So these animals are mutant for tau in all cells, but in a few percent of the cells, when you remove tau from the glia and restore the function of tau in the glia, you get a longevity increase that's pretty substantial. It's not like the controls, but it's getting very close. When she does studies where she looks at day 8 at the climbing assays, in the mutants, you see that it takes these flies much longer to climb. Many of them take eons to climb. And when she removes with flipase the gene in the glia, you fully suppress the phenotype. So that's obviously interesting because it really points to the fact that tau, at least the loss of tau, plays an important role in glia. That lipid droplets in the brain, and so here for the first time I show a brain and the lipid droplet accumulation in the brain. And what you can see here, if you express with reprogal4 the system so that you remove tau into the glia in the brain, you do not have lipid droplet formation anymore. And that is associated with elevated levels of peroxidated lipids. If you look at the other assays, when you've removed the repo only the mutant in the adult brain, or you do RNAi, as is shown here, you can see that when you do RNAi here, you see that these animals that express the RNAi against tau in the brain live shorter, that they develop a progressive climbing defect, and that they have sleep defects. And I want to point out to these sleep defects because they're really interesting. There was very recently a paper in Nature Neuroscience from the Siegel lab, and they've shown that this whole pathway of peroxidated lipids formation in neurons, transfer to glia, and formation in the glia, and consumption during the night of the peroxidated lipids in these glia is a determinative pathway in sleep. So this is an important sleep pathway, providing a physiological background to what I just told you. Sleep plays an important role, and the lipid droplets, obviously, in the sleep pathway are induced in the neurons during activity or transferred during the day to glia and consumed at night in the glia. And so I think this is an interesting example of how this pathway plays an important role in physiology. And now we turn back and we do the the RNAis in the photoreceptors to show that indeed when we knock out tau in the photoreceptors and the whole animal, so this is a full animal knockout, and again it's dose sensitive. You remove one copy of tau over a wild type and you have a dramatic reduction of the production of lipid droplets. You can fully rescue this with a genomic rescue construct, as is shown here. And then here, again, you can do, this was done with the mutant, the chromic mutant, you can do an RNAi and get the same phenotype. And finally, you can now test all the pathogenic mutants in tau. And to make a long story short, I'm not going to go through the data. When you express pathogenic variants of tau, you have a dramatic decrease of the production of peroxidated lipids, or not production of peroxidated lipids, accumulation of peroxidated lipids in glia. And so the question then is, how does tau in glia interfere with microtubules to suppress the production of lipid droplets? And the answer is and that is that if you take vertebrate cells and you load them with BODPC12, you can see that these cells will now form lipid droplets. And if you do a tau RNAi, you can see that there is apparently more. But if you look at the ER, you can notice that the ER is vastly expanded and all the lipids that are present here are present in the ER. And so if you carefully look at these data, what happens is that the ER is loaded in these RNAi for tau with lipids that cannot properly form lipid droplets. And so those cells have high levels of peroxidated lipids. If in the fly, obviously, when you do these experiments or in cell culture, because the lipids, the peroxidated lipids cannot be consumed properly. Obviously, that's an interpretation, but this suggests that the production of these lipid droplets in glia is dependent on tau. And if you manipulate microtubules in these cells, you get the same effect. It's through microtubules that these lipid droplets form. So in a summary, you have ROS, you have activation of junk SRABP, the lipids are peroxidated, they're transported to cells, they're taken up in the glia, in the ER, these lipids form lipid droplets, and then the lipid droplets are consumed. What's present in the lipid droplets as an energy source is consumed by mitochondria and may feed back to neurons. The players are involved here that are involved in Alzheimer's disease. This gene is where we started, and that is now GWAS locus for Alzheimer's disease. There's these transporters for these that are GWAS loci for Alzheimer's disease. There's apolipoproteins that are involved in this process. There is A-beta 42 that plays a role in this pathway and hijacks this pathway. And finally, there is tau that's required for the formation of these lipid droplets. So we think that this is just a summary again, but I would like to argue that given that N-acetylcysteine is such a potent suppressor in all the circumstances that we've looked at, that ROS is really at the root of this problem. And quenching ROS might be a really important strategy, therapeutic strategy in the long term. And so there's a long list of people that have played a role in this project. The MOSC, Shinya Yamamoto and Michael Wagner, and a lot of postdocs and people here. The CRIMIC and the drop-in technology, us, and a lot of other people. The Marvel, we're having a new paper coming out on Marvel with Zhangdong Liu on Marvel AI, which is really a strategy to look at patient sequences and selecting the variants that may be the cause of the disease, was done by Julia Wang and Zhangdong Liu. And the AD project I mentioned was done by Lucy Liu, Matthew Moulton, Lindsay Goodman, and other people. Obviously, you have to acknowledge all funding organizations that have supported this research. Thank you.
This concludes the Gruber program. Thank you so much for joining us today. Congratulations again to our 2024 Gruber Genetics Prize recipient, Hugo Bellen.